Okay, so we end up last time looking at the differences between the characteristics of gram-positive and gram-negative microorganisms, and that includes that gram-positives have a thick peptidoglycan layer, only one plasma membrane, and they have teichoic acids. Now, gram-negatives, they do not have teichoic acids, they have a thin peptidoglycan layer, and they have two plasma membranes. And they have lipopolysaccharide. So those are a specific characteristic of each. And then we will continue now with the mode of transport. So again, here is a picture showing you, or a couple of pictures showing you how gram-positive looks like under the microscope. So they stain purple, while gram-negatives, they stain red. So now for microorganisms that they do not stain with gram staining technique, either because they have a thick mycolic acid wall, like in the case of Mycobacterium species, or maybe they do not have a cell wall, like in the case of Mycoplasma. Or it could be that they have like a different component in their cell wall. So in this case, you call it atypical cell walls. So for instance, if you have mycobacteria, they do have a lipid that is called mycolic acid that is like a wax. And this one will be bound into the peptidoglycan. And in this case, you use the acid-fast staining technique to stain these microorganisms. And we saw this staining technique before. And one way that you can see it in the microscope is when they pick up the carbofluxine staining, which is a red stain, that it makes them to look red. And then the cells that are not acid-fast will be staining with methylene blue, which will be the counter stain. And then we have other species of bacteria that is called Nocardia, in which you also stain them with this acid-fast technique, and then they give you these same characteristics as the mycobacteria. They are acid-fast positive. Can you try to use gram staining? Yes, you can. And some bacteria, they will pick up this either safranin or crystal violet, but you will have a mixture of them. So this is called gram variable. So instead of having a gram variable, because you're using the wrong technique to stain these microorganisms, you rather use something that stain them and make them look as they're supposed to. Like mycobacterial species are definitely acid-fast microorganisms. And then Mycoplasma will be a bacteria that do not have a cell wall. So it's wallless. And this bacteria, when you grow it when you grow it in the lab, it gives you characteristic growth. So if you grow it in Petri plate, it will give you like this type of growth. It looks like fried eggs. And instead of cell wall, it has a plasma membrane. So this is a bacteria that produces infections in the lungs, and it causes what is called walking pneumonia or atypical pneumonia. Because the person doesn't feel so bad, but still has pneumonia. It has infection in the lungs. And then archaea can be characterized by having pseudomurine. So it lacks this NAM or N-acetylmuramic acid. So it has a component that's called pseudomurine instead, and it also lacks the amino acids, which is part of the amino acids that bacteria has in their cell wall, the amino acids. So some microorganisms, when you damage their cell wall, they take different shapes. They will become pleomorphic. Now, typically bacteria has only one shape, so they are monomorphic, but when you damage this cell wall, they become pleomorphic. Now, how can you damage the cell wall? Well, by adding enzymes like lysozyme. So lysozyme is an enzyme. An example where we have lysozyme, it's in saliva and tears. So then we have our own protection. So with lysozyme that we have in the saliva and the tears, we prevent infections in the eyes and infections in the oral cavity or in the digestive system in general. But it's not foolproof, of course. Whenever we have a microorganism that is very resistant to chemicals, or maybe like the lysozyme, or maybe it will have like a structural protection themselves, or it's many microorganisms. In this case, we end up with infections no matter what. And now we have this antibiotic, penicillin, that inhibits peptide bridges in peptidoglycan. So that also damages the cell wall. So if you have a microorganism, and if you add antibiotics, so you start breaking the cell wall. And then what happens is that you have cytoplasm leakage. So the cytoplasm start exiting, and then the bacteria will die. So then we use these antibiotics to treat infections. A protoplast, it's a form of bacteria that will form when it doesn't have a cell wall. And then an spheroplast is also another example, but this one, it's in a gram-positive cell, and protoplasts themselves and spheroplasts are susceptible to osmotic lysis. Like if you put bacteria in a beaker with a solution, that it has high concentration of of solutes like sodium chloride, and you put a bacteria there. So this is a hypertonic solution. Where you have high salt content. In this case, the bacteria will be under osmotic lysis. So you will make it easier to break. So it will happen something like this, and eventually it will burst. And now we have another type of bacteria that forms after you remove the cell wall, and it takes an irregular shape, and you call it an L form. Now, an L form is not related to taking the L letter shape. It's related to actually the institute name, the first letter of the institute name where it was discovered, which was the Lister Institute in France. Joseph Lister was a physician who started to use some disinfectants when performing surgeries, and then they named several things in his honor, including this institute in France. That's a research institute. So this L form, it's a wall-less bacteria that can take irregular shapes, and that it swells. Okay, so then let's look at the components of the plasma membrane, and this is supposed to be familiar to you either from AMP1, AMP2, or both, or biology classes. So phospholipid bilayer is the component of the plasma membrane that is more abundant, and it is made out of two leaflets of phospholipids, and then you have the fatty acid tails facing medially away from water, so this will be hydrophobic, and then you have phosphate groups, that's why it's called phospholipids, because it has lipids that faces medially, and then the polar heads or the charged head of the phosphate groups, they face towards internal environment in the cell or towards the external environment in the cell. So they can interact with water, aqueous environment, so they will be polar. So they will be hydrophilic. And then embedded within the plasma membrane, you have peripheral proteins. Proteins that will be attached to either of the leaflets of the phospholipids. And then you can have proteins that are integral or transmembrane that can go across the two leaflets of the plasma membrane. And you can have other proteins that have different functions. In the case of bacteria, it will be channels, for instance, and also in eukaryotic cells. So here is the characteristics then of this plasma membrane, which will be, in this case, a gram-negative because you have two plasma membranes, the inner and the outer. So here is the inner and here's the outer. So if you magnify then this plasma membrane, you can see these phospholipids. If you magnify the phospholipids, this is how they look. They have two fatty acid tails, so this is one phospholipid, this is another one. This is from the outer leaflet, this is from the inner leaflet. So here you have two fatty acid tails, this is a phosphate group. And then here you have some integral proteins. In this case, this is the channel that it has this little gate internally. It's not open all the time. And then you have some proteins that can attach to these integral proteins, and this will be called peripheral. And this portion of the phospholipids, this lipid layer, will be hydrophobic, while this one will be hydrophilic. That's the polar head, the phosphate group. It has a negative charge. Any questions? So what happens in this plasma membrane? Well, you have permeability. So this plasma membrane is not completely sealed. It has some openings that allows the passage of molecules. In this case, will be channels that allow the passage of ions, nutrients, and then you can have aquaporins that lets water to come in and out. And through the phospholipid layers, you can allow the passage of molecules like CO2 and oxygen, which will be diffusing without the aid of a channel because these are basically very small that they can traverse these phospholipid bilayers. Now, not everything can go through this plasma membrane. Only certain things, like for instance, in sodium channels, only sodium. In potassium channels, only potassium. And then you have sometimes embedded within this plasma membrane enzymes that allows the production of ATP so that the cell can have energy. And some of them, they can have some pigments, photosynthetic pigments, that allows the creation of photosynthesis, and they're found in foldings that are called chromatophores or thylakoids in the case of plant cells. So here we have transmission electron micrograph of a gigantic rod, in this case, with chromatophores. And that will be gigantic, of course, in the microbial world in comparison to other cells. And then you can see here in the surface these chromatophores with pigments. Internally, these are the pigments that allow the production of photosynthesis. So in a photosynthesis reaction, this is what you will have. You combine CO2 and water, you have light, and then you will create carbohydrates plus oxygen. So that's photosynthesis. And then underneath the outer membrane of gram-negative bacteria, you have a space that is called periplasmic space. And this periplasmic space, it is filled with enzymes that helps for the bacteria to prevent phagocytosis. So what is phagocytosis? Will be eating. So this bacteria that is gram-negative prevents the action of the phagocytes, the cells that can destroy them. Also prevents the destruction by a series of proteins that many host cells have. That is called the complement system. Like our body, our immune system has a series of proteins that is called complement. We will see that later. And then also it helps to destroy antibiotics. Now, some bacteria has this O polysaccharide that it is useful to distinguish certain species of bacteria, including E. coli. E. coli O157H7 has an O antigen. It's called 157. And then in the case of gram negatives, you also have lipid A, which is the endotoxin. So what does endotoxin do? When they are released into the bloodstream. So it says the temperature center in the hypothalamus, and then the person will have fever. So that's the action of the endotoxins, and it's lipid A. So we have a reaction when someone has, for instance, Neisseria gonorrhea infection, and the person receives a shot of penicillin or an antibiotic to kill this bacteria. This bacteria is a gram negative. So this bacteria will release, when it's getting killed by this antibiotic, will release lipid A. And the person will have fever after taking this antibiotic. So there is this reaction, it's called Hersheimer, and then you have to warn the patient. After you got the shot of penicillin or ampicillin, whatever drug you give, or you take this antibiotic, you have this reaction, it's normal. Okay. So they will have fever. And then you have as well, within this space, porins. That allows the passage of nutrients and also some items in and out of the cells. So that's characteristic of this outer membrane and part or this periplasmic space. Now, for the gram-positive, they have only one plasma membrane, and then this plasma membrane will have lipoteichoic acids, which are basically very characteristic of this gram-positive bacteria. And then you have also teichoic acids. So if someone does a molecular study and then they find these lipoteichoic acids and teichoic acids, unequivocally the person will say, well, this is a gram-positive microorganism. And this is a reminder of what you have seen before about the difference between a gram-positive cell wall and a gram-negative cell wall. So again, the gram-positive cell wall is thicker, the wall. It's made by 20, 30 layers of peptidoglycan, and it has embedded to them teichoic acid in its wall, and underneath it has this plasma membrane that has also teichoic acids, but in this case would be called lipoteichoic acids because it is embedded or attached into this plasma membrane. And then you can see these cross bridges with these polypeptides, and you have the tetrapeptides. In the case of the gram-negative cell wall, it's very thin, 1 to 3 layers, and it's found here in between the outer membrane and the plasma membrane. And in this outer membrane, that's where you have the LPS, lipopolysaccharide, that has this lipid A portion. It has this core polysaccharide, and at the tip, it has the O polysaccharide or the O antigen. That in some cases, it helps for this bacteria to be identified with the tip of this LPS, the O antigen. And then this lipid A, the one that is embedded in this leaflet of this outer membrane, is the one that resets the thermoregulatory center in the hypothalamus, so we end up with fever. So just as we can damage the cell wall, we can damage also the plasma membrane. Plasma membrane is easily damaged with solvents like alcohol, quaternary ammonium, which are detergents, or with antibiotics like polymyxin. In the case of the cell wall, you can use penicillin to damage the cell wall. But in the case of the plasma membrane, you can damage it with polymyxin, which is a type of antibiotic. Of antibiotic that it is found over the counter. It's an ointment, actually. And then what you do when you damage the plasma membrane, you create pores, and also that leads to leakage of the cell content. So it's like now you have here your cell wall damage, and then you have internally your plasma membrane. So this is the plasma membrane. And now you add this antibiotic, polymyxin. So plasma membrane damage by polymyxin. Polymyxin B. Now, functions of the plasma membrane. Again, it is a selectively permeable barrier. It separates the cells from other cells and maintains the cytoplasm components in place. So what happens? So you can have movement of solutes from an area of high concentration to an area of low concentration. So from more to less. So what you can see here is a magnification of a piece of plasma membrane with a phospholipid bilayer here. And then these spheres represent solutes, particles. And these particles, as you can see here from the label, are more concentrated out. So in the top of this plasma membrane represents the outside of the cells. So they are more concentrated outside than inside. Inside you have very little. How many particles are out? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35. More or less it's like 35. And how many we have inside? How many did you count? 11. So the molecules will move from 35 to 11. And in this case will be until you reach an equilibrium. 35 plus 11 will be 46, right? What is 46 divided by 2? 23. How many molecules will move from outside to in to make it even on both sides? 12. So you will reach an equilibrium when 12 of these molecules will move here. That's simple diffusion, moving from more to less. Classical example is having a beaker with water. You drop into the bottom of the beaker a pill with colorant, and then this colorant will start diffusing from more, which is the bottom, to less, which is on the top, until all the dye from the colorant, it just keeps this water to be equally colored. So that was simple diffusion. Now for facilitated diffusion, so you will have solutes combining with a transporter. In this case, we'll be combining with this channel. And you can have two types of transporters, non-specific and specific. In the case of the non-specific transporters, they can allow the passage of molecules as long as they have a particular shape and maybe a charge. The size that it won't be as big, the size that it won't be as big, but as long as it can fit through here, through this pore, they can cross. So that will be non-specific because the molecules can cross without having so much trouble. Now, in the case of a specific transport, well, you have a gap within, or a space within these channels that allows the insert or the physical contact of a molecule that fits specifically into that space. So in this case will be, for instance, glucose. This can be a glucose transporter. And there is this space on the top of this channel that allows this glucose to fill in here, and then the channel will open in this space, and then it will free up the glucose. So that's facilitated diffusion. Now, in the case of osmosis, osmosis is the transport of water from an area of high concentration of water to low concentration of water, or from an area of high concentration of, from low concentration of solutes to high concentration of solutes. So the purpose of water movement is to allow to have isotonicity, basically having the same concentration of ions between compartments. And then water will stop moving when you reach that tonicity or that concentration of solutes. So the osmotic pressure is the pressure that is needed to stop the concentration of water. Sorry, to stop the movement of water. So the typical exercise or example is having a beaker, and then this beaker has water with no solutes. and then you introduce a glass tube with a cellophane bag inserted into this glass tube, one end, and then you have a rubber stopper that is sealing this bag of cellophane. Cellophane is like a very thin film that sometimes is used to, for instance, to wrap candy, hard candy. So then in this case, this cellophane bag has water and also has solids in them. And then it's sealed in here to prevent these solids to escaping into the glass. So then when you introduce this little bag that looks like a pouch with this solution of, for instance, sucrose, you insert it into this beaker with water. Water will move from low concentration of solids. In this case, you don't have any concentration of solids, so from high concentration of water or low concentration of solids to high concentration of solids, which are found within this sucrose solution in this cellophane bag. So water will keep moving, filling this little pouch made out of cellophane more and more until no more water will move, because you have reached equilibrium. So then you have equal amount of water between this cellophane bag and this beaker, and then the water will rise through this tube up to this level. So you stop moving more water. So this is osmotic pressure. So that's when you reach equilibrium. Any questions? So then something similar can happen to bacteria. Bacteria in its cytoplasm has a specific concentration of solids. And these bacteria can encounter two types of environment: isotonic or normotonic. It can encounter a hypertonic, or it can encounter a hypotonic environment. And depending on where it is whether it will lose water or it will remain with the same amount of water. Because the cytoplasm itself, it has a particular amount of water. And then we are thinking that the bacteria doesn't have any problems, like the plasma membrane or the cell wall is not damaged. So then you can have isotonic solution, hypertonic solution, or hypotonic solution. Hypertonic refers to high, hypo to low, iso same. So what type of concentration of, let's say sodium chloride, you have? Well, in an isotonic solution, you have 0.85% sodium chloride plus 99.15% water. Hypertonic solution, it will be, let's say, greater than 0.85, so it will be, let's say, 0.86 sodium chloride and above, of course, plus 99.14% of water. Hypotonic will be lesser than. So it will be lesser than 0.85, so let's say it's 0.84%, and below sodium chloride plus 99.16% of water. So in hypertonic, you can have like 1% of sodium chloride, 3%, 5%. That's hypertonic. So something that you can see here in isotonic is that your bacteria will have movement in and out of its cells of water, but the volume, it doesn't change. And you have, of course, to have movement of water equally in both directions because the bacteria, can excrete wastes combined with water or can introduce nutrients dissolving water. But no net movement of water occurs. Now, in hypotonic solution, like when you have very little solutes, these little spheres, brown spheres, represent solutes. In this case, you have very little solute outside of the cells. So then water will move from high water concentration or low solute concentration to more. So in this case, these spheres these solutes are more abundant inside the cells. So then the watering effort to dilute this hypertonicity will move into the cells and the cells will burst through osmotic lysis. In a hypertonic solution, when you have high concentration of solutes with respect to the cytoplasm, like see, very little solutes in comparison to here, then water will move from low solute concentration to high solute concentration, or from high content of water to more content of water to try to dilute this, and the cells will shrink. So this is plasmolysis. In this case, the plasma membrane, see how the plasma membrane separates from the cell wall. And this separation will lead to a dead signal, so the cells will die. So any questions? Now, we also have aquaporins. In this case, the aquaporins are channels that are specific for water. Aqua means water. That's the Latin for water. So water will move through aquaporins. And then we also have active transport. In active transport, you would use energy, ATP, and you require a transporter to transport molecules. So you require a transporter and you require ATP. And then you have a type of movement that is called group translocation, in which as the molecule is going through these channels, you can change the molecule. In this case, for group translocation, you require phosphoenol pyruvate and also a transporter, and that leads to the movement of molecules. Phosphoenol pyruvate is also known as PEP, PEP, or PEP. So those were then components of the plasma membrane and their function, the channels, how they help for transporting molecules. Now let's talk about the internal components of the bacteria. So cytoplasm. Cytoplasm is the substance that is located inside the plasma membrane, and it helps to hold ribosomes and the nucleoid, which is the molecule that is the DNA in the case of bacteria. So here we have then the cytoplasm as seen in this transmission electron micrograph, and this is the nucleoid that has the DNA. Ribosomes are the protein factories of the cells. And then in the case of prokaryotes, they have a ribosome that is called 70S. And it has two subunits, or two small parts. So it has one that is the large subunit that is called 50S, and then it has a small subunit that is called the 30S. And then both of them get in contact like this, and they form the complete ribosome. So through this little tunnel, these ribosomes read the messenger RNA, and they produce a protein. Now, 70S stands for a unit of sedimentation. It's called Sedberg. And then they don't add arithmetically. It's just a measurement of how quickly they go into the bottom of a test tube when you centrifuge at a high speed samples that contains ribosomes. So this one is heavier than this. Now, in the cytoplasm, you not only have the nucleoid, but also you have inclusions or storage units. So these inclusions can have several items, including phosphate reserves that are called metachromatic granules or volutin. You can have glycogen. In this case, will be called polysaccharide granules. You can have lipids. So in this case, you call it lipid inclusions. You can have sulfur, and you call it sulfur granules. And then you can have some that has an enzyme that helps for obtaining CO2 and fix it. This is called carboxysomes. And the enzyme that they have is ribulose 1,5-diphosphate carboxylase. Some of them, they have inclusions with gas. In this case will be called gas vacuoles, and they look like cylinders. And this gas, as they accumulate or they release it, helps them with buoyancy, which is floating in aquatic environments. So they can go up or down the level of water with these gas vacuoles. And some of them, they can have metals like iron oxide. So these will be called magnetosomes. And they use it to destroy hydrogen peroxide. more than likely have seen hydrogen peroxide in bottles. So it's a molecule that is helpful to destroy microorganisms. And some microorganisms can defend themselves using these magnetosomes. So here we have a rod bacteria that has two polar flagella. So it's Amphitrachus. And then it has in this middle part these magnetosomes. So these are vacuoles filled with iron oxide. And they enhance the color to show you how potentially will look like reddish. Iron in certain oxidation state gives you like reddish color. So this is a transmission micrograph with this bacteria. Now, some bacteria, they produce endospores. So, Bacillus species So these are bacteria that produce endospores. Endospores are resting bacteria. They're not the vegetative form. I told you before, when do they form? They form when the bacteria is in an environment where you don't have nutrients, maybe you're running out of water, maybe it's under the attack of radiation. So these endospores are resistant to desiccation, lack of water, high heat, and chemicals. When bacteria produces these endospores, this is called sporulation. And then these seed into the environment when they find like water or nutrients or a better environment, better temperature, and they form a new bacteria. And in this case, it's called germination or return to a vegetative state. So this is transmission micrograph showing you an endospore. So this is where you have the DNA, and then here you have the components of this endospore that protects the DNA. In this case, this is from Bacillus subtilis. And this is the process of sporulation. So first you have to make a copy of DNA. So you duplicate your DNA. And then you start forming a spore septum here to isolate a newly replicated DNA in a small portion of the cytoplasm, which is this. After the plasma membrane surrounds the DNA, and you isolated this DNA, you will start forming a second membrane. And this is going to be called a forespore. After this, you introduce a cell wall between the two membranes, and then you form a coat outside of this membrane, and you release the endospore. So, curiously enough, you have two plasma membranes and a peptidoglycan layer. What does this remind you of? What type of bacteria, gram-positive or gram-negative? Because you have two plasma membranes. It's only one or the other, gram-positive or gram-negative? Gram-negative. So it looks like a gram-negative, but it's actually coming from a gram-positive bacteria. Okay, now let's move into animal cells. So, components of the animal cells: a nucleus surrounded by a nuclear membrane. You have nucleoplasm, you have a nucleolus. Within this nucleoplasm, you have DNA, and then you have these pores of the nuclear membrane that allows messenger RNA exit the nucleus. And then attached to this nucleus, you have this rough endoplasmic reticulum, which has these ribosomes attached to them to produce proteins. And then you have a Golgi apparatus, which is this, and then you have a smooth endoplasmic reticulum, you have mitochondria, you have vacuoles, you have lysosomes, you have free ribosomes, centrioles, and proteins that forms a structure that is called the cytoskeleton. And then you have a flagella if the animal cells has a flagella, and then you have this plasma membrane. If you have an eukaryotic cell that has a cell wall, it can be fungus or it can be a plant cell. In this case, this is a plant cell, and you have a chloroplast, and you have a large vacuole. And the other components similar to the ones from the animal cell. So this is transmission electromicrograph of plant cells and animal cells. So you have peroxisomes here, mitochondria, chloroplast. You have Golgi complex here, cell wall. This is actually an algal cell, Tribonema vulgaris. This is an algae. And then for animal cells, you have nucleolus here. This is the nucleus. And you have the cytoplasm in here. You have mitochondria, lysosomes, rough endoplasmic reticulum, etc. So this is an actually antibody secreting plasma cell. So this is a lymphocyte. So flagella and cilia. So in this case, you have Euglena, which is a chlorophyll containing algae, and it has a flagella. Now here you have a protozoan, freshwater protozoan, which is called Tetrahymena, and it has cilia everywhere. Now, if you look at the movement of flagella, it can be undulant, like a wave, or it can go sideways, it can rotate. So this Euglena can use the flagella by rotation or by undulation. Now, if you make a section of this flagella and you see it internally, flagella is surrounded by a plasma membrane, and internally you have microtubules made out of protein that is called tubulin. These microtubules form pairs around two central microtubules. So this is called 9+2 arrangement. So this will be 2 in the center and 9 pairs on the periphery surrounding these central microtubules. In terms of the cell wall of eukaryotes, they are simpler than the cell wall of prokaryotes. And then what eukaryotes have cell wall? Plants, algae, and fungi. And these cell walls are mainly made out of carbohydrates. Now, in the case of algae and fungi, these carbohydrates will be cellulose and chitin. Yeast will have glucan and mannan. Now, just as the prokaryotic cells, we can also have glycocalyx, which are the carbohydrates that covers the plasma membrane in animal cells. And these glycocalyx will have glycoproteins and glycolipids that helps for adhesion of these glycocalyx. Plasma membrane in animal cells or eukaryotic cells. You have also phospholipid bilayer, which creates a selectively permeable membrane. You have peripheral proteins, integral proteins, transmembrane proteins, and you have a molecule in certain eukaryotic cells like esters. Like in animal cells, you have cholesterol, and then in fungi, you have ergosterols. And then you have glycocalyx carbohydrates. So mode of transport is similar to prokaryotes. You have simple diffusion, facilitated diffusion, osmosis, and active transport. Active transport, you require ATP. And in the case of ingesting materials, in the case of eukaryotic cells, you call it endocytosis. And endocytosis will be phagocytosis when you extend pseudopods or false feet. You surround whatever material the cells is going to ingest or eat, and then they bring it in. So they engulf it. And pinocytosis will be for the same process, but in this case, instead of being a solid, it's going to be a liquid. So you bring dissolved substances within this liquid in pinocytosis. Eukaryotic cells have a cytoplasm that has cytoskeletal proteins of different types. So you can have actin, myosin, tubulin. And then the fluid portion of the cytoplasm is going to be called the cytosol. And then within this cytosol, you can have streaming to help to distribute the nutrients equally, or whatever is found within this cytoplasm. You can move it through this streaming, like creating little waves. And then you have enzymes that are stored in organelles, like in lysosomes. And then in the prokaryotes, those enzymes are not found within these vacuoles. So here is a magnification of this nucleus. So you have a double nuclear membrane with pores, which are this, and then you have the nucleolus, and then the chromosomes are found within the nucleoplasm, and this is called the chromatin. So it's the DNA in the nucleus, the chromatin. So then you have your chromosomes containing the DNA. And the plasmic reticulum, you have two types: rough, that helps you to produce proteins, and then you have smooth, that helps you to produce lipids. So that's where you can make plasma membrane. And the difference is that the smooth doesn't have the ribosomes. So this is a magnification using transmission electron micrograph. So you see the lines, which is basically a sack of membranes. And this one, since it doesn't have like these little studs, which are the ribosomes, that's why it's called smooth, while this one is called rough. And inside the rough, the messenger RNA coming from the nucleus goes through, and then you start reading this messenger RNA. So, then you have processes in biology. So you have transcription, you have duplication, and you have translation. So if you make a copy of DNA out of DNA, that it is duplication or replication. If you make a copy of messenger RNA out of DNA, that is translation. And if you make a protein out of messenger RNA, that is translation. Sorry, transcription is making a copy of messenger RNA out of a DNA template. So ribosomes then are where translation takes place, and these are larger and denser than the prokaryotic ribosomes. And they can be attached to the rough endoplasmic reticulum or can be found free in the ribosomes. So we have 70S ribosomes in chloroplasts and mitochondria, but in eukaryotic cells typically is 80S. So we will stop here. Do you have any questions?